Hello guys, welcome to Big Book. Today we are going to discuss a new topic about enhancer in genetics. Before to that, I wish to you people to look into these images. This is the transcription template where you are finding number 1 here, this to be as the promoter region, and number 2, this to be as the expression region of the gene, and number 3, this to be as the enhancer region. Number 4, this to be as the insulator region, and number 5, this is the transcription factor, and number 6, this is the dimer. Number 7, this is the enhancer factors. These transcription factors, the dimers and enhancer factors, they are going to become a complex of the factors where we are going to study the enhancer in the proper way. And this is how the transcription DNA is going to change its structure, forming a loop during the enhancer expression. So this is the total complex of the dimer, transcription factor and the enhancer factor, and this complex is also called to be as the activator proteins or the activator factors. Now, enhancer, it is a nucleic acid sequence that increases the transcription or utilization of a gene. Here it is a small nucleic acid sequence which will be present within the DNA sequence, where it can be up to the upstream or the downstream of the DNA gene. Here you can see that the increase in the transcription means the number of the fold of the transcription of that particular gene is going to increase. It is many more number of times the transcription is going to takes place to give the mRNA molecule, where the mRNA molecule further takes place, that amount of the mRNA molecule, that number of the mRNA molecule further going to take place in the translation process. So it is again meant into the form of the utilization of a gene. Again, the enhancer is a DNA sequence present within the control regions of many eukaryotic genes that can enhance transcription. This is the other form of the definition of the enhancer. The enhancer can generally function even if inverted or if placed in a different position some distance from the gene it controls. Activation of the enhancer requires certain regulatory proteins. Here regulatory proteins is nothing but the enhancer activation proteins and the transcription factors, and also the dimers, which has been already shown in the picture. A nucleotide sequence that increases the rate of genetic transcription by preferentially increasing the activity of the nearest promoter on the same DNA molecule. So this is also the same other words, but the meaning is the same. Here particularly one thing has been mentioned in this definition, that is a enhancer will be present for a DNA expression. In the sense, if a gene is expressible gene is present, that expressible gene will be having its own specific kind of the enhancer. So that particular enhancer will be responsible for the manifold of the transcription of that particular gene. This is the meaning of it. Now here we will discuss about enhancer, how it is. Enhancer is of 50 to 1500 base pairs. This is the minimum or minimum base pair sequence length of an enhancer. the region of DNA that can be bound by proteins. So already we have spoken about the activators to increase the likelihood that transcription of a particular gene will occur. These proteins are usually referred to as transcription factors, already spoken. Enhancers or cis-acting. Enhancers or cis-acting. They can be located up to 1 million base pairs away from the gene, upstream or downstream from the start site. So as we have already spoken that the enhancer can be present on the DNA sequence at any position or any location. So it can be far away. Minimum distance of the enhancer on the gene can be up to 1000 million base pairs. That can be on the upstream of the gene or the downstream of the gene, and the same distance can be between the distance can position between the introns of the gene also. So this is how the enhancer will be present. So enhancers, some transcription factors bind to these enhancers. These are also called as enhancer binding proteins. So here, binding to regions of DNA that are thousands of base pairs away from the gene they control. Binding increases the rate of the transcription of the gene. So here already we have spoken about the loop formation. Here you can see this is the DNA expression loop. This is the promoter. This is the enhancer. Okay, and again here is the exon. So here from here the promoter, and this is the exon. So the promoter has been activated. So further here from here the DNA polymerases are going to take part in the transcription process. So the transcription is going to pass on the DNA, which is expressible. Further the exons and introns, and further the introns will be removed by the post-transcriptional processes. Further the procedures, you know how the translation is going to takes place. Now here how the loop is formed. Enhancers can be located upstream, downstream, or even within the gene they control. This we have already discussed. There are thousands of enhancers in the genome, but which ones are active depends on the type of the cell and the signals which it is receiving. Most genes, at least in Drosophila, are regulated by 2 to 3 gene enhancers, but some may be controlled by 8 or more. Multiple enhancers are particularly characteristics of housekeeping genes. Here in this description, the first part is related to depends on the type of the cell and the signal which it is receiving. We have heard about the induction and inhibition process within the developmental of the cell or the expression of the cell. So here both development of the organism, during the development of organism, and the expression of the organism. So in induction, in the sense a cell signal during the developmental process, when two different cells has been developing, one cell will be having the different type of its expression according to its tissue, and the kind of the enzymes, proteins, and the hormones should be synthesized in that particular cell that is be synthesized. The neighboring tissue will be having the different expression compared to the that particular next neighboring cell. So whatever the induction cells induction signals has been shared between these two different tissues, those are going to form the induction signals to the other tissue where it is performing the different type of the signal, different type of the gene expression. So it is called to be as the induction, and also the same signal is going to act as the inhibition signal, where it is going to call that particular neighboring tissue or the tissue cell not to produce the same kind of the protein or the enzyme or the hormone which is produced by this particular signal sending cell or the tissue. So here this is what it is meant to be. Which ones are active depends on the type of the cell and the signals which it is receiving. Okay, so most genes, at least in Drosophila, are regulated by 2 to 3 enhancers. So here most genes will be having the 2 to 3 enhancers. Okay, where in Drosophila we will study the development of the uh, embryo of the Drosophila or the Drosophila egg. During that time we can observe the 2 to 3 enhancers will be active to produce the repeated number of the mRNAs, where this repeated number of the mRNAs will be responsible for the protein synthesis for the further development of the organisms. So it has also been seen that it is controlled by the 8 or more enhancers. So again, there is multiple enhancers or particularly characteristics of housekeeping genes. Housekeeping genes in the sense, for a particular tissue, a particular form of the protein, a particular form of the enzyme, and a particular form of the hormones, and a particular form of the structure of the cell, and a particular form of the inhibition and the induction signals has to be produced. So that particular group of the genes will be always be active. This particular group of the genes which are always being active is called to be as the housekeeping genes. These housekeeping genes will be having the enhancers which are always active. Okay, so this will be having the enhancer which are always active. Those housekeeping genes will be having the multiple enhancers in particular. So this is what meant by this particular. Now we will discuss how does the binding of a protein to an enhancer regulate the transcription of a gene thousands of base pairs away. One possibility is that enhancer binding proteins, in addition to their DNA binding site, have sites that bind to transcription factors assembled at a promoter of the gene. So here already we have seen in the particular in the picture that the formation of the loop has been taken place, where the formation of the loop has given the binding of the transcription factors, enhancer activating factors, or the dimer formation, okay, which is going to form the complex between a promoter and a enhancer. So this is the assemblance is going to induct the promoter of the gene. So here how it is going to form, this would draw the DNA into a loop form as shown in the figure which we have already seen. Now here there is a visual evidence of the same, which is by the Mitchell or Botchen, who kindly supplied these electron micrographs, and his colleagues have produced visual evidence of this model of enhancer action. They created an artificial DNA molecule. Here this upper picture is one which is not marked one. This below picture is marked one, which is showing a loop within the yellow circle. Okay, and a complex which has been formed is shown by the pink arrow marks, which is blue and red. Okay, and 1860 base pairs of the enhancer loop has been shown. So here this is the particular enhancer loop formation, which is the electron micrograph formed. Significance of looping. Looping of chromosomes that brings enhancers close to promoters and promoters close to other promoters. So here there can be two promoters also. In some genes there will be present of the two promoters, where both the promoters has to certain times both the promoters are to take part in the expression of the gene or the formation of the mRNA or the DNA transcription takes place, where seems to be a mechanism to ensure the expression or inhibition of groups of genes that must perform together. Here, already, as we have already spoken, that expression or inhibition of groups of genes has to be taken part. In certain gene sequences, two promoters will be responsible for two different transcriptions. Promoter 1 will be, the first promoter will be responsible for the first different kind of the mRNA sequences of the same DNA sequence which is in the up downstream of that particular promoter, and the same way the promoter 2 will be responsible for the different mRNA sequences to be expression. So here that can also be happened. The response of a cell to the arrival of a signal may involve turning on or off hundreds of different genes whose products must be produced in a coordinated way for the cell to respond appropriately. To respond appropriately, the dynamic movement of the portions of the chromosome carrying the appropriate gene loci into a transcription factory may be a mechanism to accomplish. Here, these words are giving a very clear meaning of formation of the loop, where the enhancer is going to move to that loci of the DNA molecule. Here they have used the word chromosome, don't get confused. It's a DNA where the enhancer DNA sequence is going to come to the promoter region, where the complex of the transcription factors, or altogether the transcription factors, dimer formation, and the enhancer activator proteins, they together called it to as the transcription factory, where this is going to form the complex, and further the whatever the mRNA is going to be synthesized, that is going to be uh, takes place with the help of the RNA polymerase enzymes. Examples in human genome is HSCNS1, also known as CERTG2, and located in the human accelerated region 2, is a gene enhancer that may have contributed to the evolution of the uniquely opposable human thumb, and possibly also modifications in the ankle or foot that allow humans to walk on two legs. Evidence to date shows that of the 110,000 gene enhancer sequences identified in the genome, HSCNS1 has undergone the most change during the evolution of humans following the split with the ancestors of chimpanzees. And one more example is an enhancer near the genes GADD45G has been described that may regulate brain growth in chimpanzees and other mammals, but not in humans. The GADD45G regulator in mice and chimps is active in regions of the brain where cells that form the cortex, ventral forebrain, and thalamus are located, and may suppress further neurogenesis. Loss of the GADD45G enhancer in humans may contribute to an increase of certain neuronal populations and to forebrain expansions in humans. Dimer is a chemical structure formed from two similar subunits. Protein dimer, a protein quaternary structure. Thank you for watching the video, and be tuned to the big book. We will come with new topics. Thank you very much.